Welcome. This explainer provides a mechanistic review of tumor suppressor genes, structured specifically for the CSIR-NET Life Sciences examination.
We'll be covering 5 sections. We'll start with the definition, move to normal functions, then discuss inactivation mechanisms, the two-hit hypothesis, and finally, look at the key examples of RB and P53.
Okay, let's begin. First, we need to establish a precise definition for tumor suppressor genes.
So, by definition, these are genes whose normal function is to inhibit cell proliferation. This means that a loss-of-function mutation, one that makes the gene product non-functional, is what contributes to cancer. They are the negative regulators of cell growth.
A few core properties to remember here. First, their action is generally recessive at the cellular level, meaning both copies of the gene must be inactivated. Second, the mutations are categorized as loss of function, resulting in a non-functional protein. And third, their role is fundamentally inhibitory. They restrain cell growth and maintain stability.
Let's move on to the normal cellular functions of these genes. What exactly are their physiological roles?
We can group these proteins into functional classes. Some, like APC and PTEN, are inhibitors of mitogenic signaling pathways. Others, such as RB and P16, are direct regulators of the cell cycle, imposing critical checkpoints. A third class, including TP53 and the BRCA proteins, maintain genomic stability by responding to DNA damage. And finally, some, like E-cadherin, maintain cell-cell adhesion.
So, to summarize, their activities fall into 5 major categories: regulating cell cycle entry, receiving anti-proliferative signals, acting as DNA damage checkpoints, promoting apoptosis, and participating directly in DNA repair.
Now, let's examine the specific molecular events that lead to the functional loss of tumor suppressor genes.
Inactivation can occur through 2 primary pathways. You have genetic mechanisms, which involve direct changes to the DNA sequence itself, like mutations or deletions. Then you have epigenetic mechanisms, which silence gene expression without altering the DNA sequence, for example, through promoter hypermethylation.
Loss of heterozygosity, or LOH, is a key genetic mechanism. This describes the event where a cell, which is already heterozygous for a mutant allele, loses the one remaining functional allele. This is a very common way the second hit occurs.
And how does LOH happen? The 2 primary causes are somatic events during mitosis. Mitotic recombination between homologous chromatids can result in a daughter cell that is homozygous for the mutant allele. Alternatively, chromosome missegregation can lead to the complete loss of the chromosome carrying the normal allele.
The other pathway, epigenetic silencing, achieves inactivation differently. Here, the gene's DNA sequence is unchanged. Instead, modifications to the chromatin, like DNA methylation in the promoter region, lead to transcriptional inactivation. The gene is effectively turned off, and this silent state is heritable.
All right, this brings us to the canonical genetic model that explains the inheritance patterns associated with these genes.
This is the two-hit hypothesis proposed by Alfred Gudson. It states that 2 separate mutational events, or hits, are necessary to inactivate both alleles of a tumor suppressor gene, which is a required step for tumorigenesis.
Retinoblastoma provides the classic example. In the familial form, the first hit is an inherited germline mutation present in all cells. This means only a single additional somatic mutation is needed for a tumor to form, resulting in a high probability of multiple tumors. In sporadic retinoblastoma, 2 independent somatic mutations must occur in the same cell, which is a much lower probability event, typically resulting in a single tumor.
Finally, let's apply these concepts by examining the 2 most important tumor suppressor genes, RB and P53. First, the retinoblastoma gene, RB. This was the first tumor suppressor gene discovered, and it serves as the prototype for this class of genes due to its central role in cell cycle control.
Mechanistically, the RB protein is a negative regulator of the G1 to S phase transition. When it's in its active hypophosphorylated state, it binds and inhibits E2F transcription factors. This prevents the cell from progressing into S phase. To proceed, cyclin CDK complexes must phosphorylate RB, inactivating it and releasing E2F.
Next is TP53. This gene is the most frequently mutated gene across all human cancers, appearing in over 50% of cases. Its protein product, P53, plays a central role in maintaining genomic integrity.
The P53 protein is a transcription factor that acts as a sensor for genotoxic stress.
When activated by DNA damage, it induces target genes that lead to several outcomes, most notably cell cycle arrest through the induction of P21, as well as DNA repair, and, if damage is too extensive, apoptosis.
Now, an important exception with P53 is the concept of a dominant negative mutation. Because the P53 protein functions as a tetramer, a mutation in just one allele can produce an altered protein subunit. When this faulty subunit is incorporated into the tetramer, it can render the entire complex non-functional. This explains how a mutation in a single allele can have a dominant effect at the cellular level.
To conclude, the inactivation of tumor suppressor genes is a requisite step in tumorigenesis. Understanding these molecular mechanisms, from genetic and epigenetic inactivation to the functional roles of key proteins like RB and P53, is essential for a comprehensive knowledge of the molecular basis of cancer.